Welcome to the PharmaSource podcast.
Most promising antibody therapeutics don't fail because the science is wrong. They stall because the infrastructure, expertise and execution support simply aren't there at the right time. For early stage biotech teams and academic groups, that gap between a validated target and a phase 1 trial can feel insurmountable, and it often is without the right help. My guests today are Debby Vermeer and Richard van Rijnsoever. Together they co-founded ExpediteBio, a new venture focused specifically on closing that gap for early stage biologics programs. Debby also co-leads the Biologics workstream at Oncode Accelerator, a national initiative in the Netherlands working to build the infrastructure that gets cancer therapies to patients faster. In this episode, we get into where development programs actually break down, what it takes to build the right partnerships, and how academic teams can navigate the transition into serious drug development, and what a better functioning biologics ecosystem actually could look like in practice. Thank you for joining me, Debby and Richard. First, tell me a little bit about yourself. Debby, do you want to start with you? Tell me a bit about your background.
I have a background in therapeutic antibody discovery and development in the CRO space, so contract research organizations. And with that, I served different roles from scientific to commercial ones. So after finishing my PhD, I started as a scientist, generating antibodies for our clients, actually hands-on myself. And quickly thereafter, I was already very interested in that client mindset. What do they need? How can we bring the products that they want in their therapeutic program? So I quickly became a project manager to guide that, and eventually more into the business development roles, etc. After which I took the role of the head contract research organization because of a reorganization. So that was actually the start of the management part. And prior to becoming an independent advisor, I served the role as general manager of that CRO site. And that was because the site got acquired by a Canadian company, and a Dutch general manager was required for that. So already said that afterwards I started as an independent advisor, mainly active information of larger public and private alliances and collaborations, and advancing smaller biotech companies into that alliance or supporting them with business development approaches. So two different activities, supporting smaller companies and within the larger Dutch and European landscape, find ideal partners to create an alliance. And since December 2025, Richard and I co-founded ExpediteBio, where I lead the position of eSite Strategic Alliance Officer.
And Richard, how about you? How did you get where you are today?
Yeah, Richard van Rijnsoever, and I've got a background in biologics development. So one started as a scientist at the lab at a company called Introgene, which later became Crucell. Did a lot of work on gene therapy and all kinds of vectors that were needed from that stage. But since the science, I actually also moved to what we sometimes call the darker side, which is business development. And I did that at a lot of biotech companies and later on also in the service environment. So at CDMOs, where I was supporting programs basically from early discovery stage to clinical development and even into market supply. And I've spent most of my career on that edge of science and the execution of getting it into the clinic and to the market, and helping to translate these companies from making that step by connecting strategy, CMC, and the actual practical execution. And as Debby mentioned, we recently founded ExpediteBio simply because both Debby and I have spent a lot of years in this industry, but we kept seeing the same issue here. And that was that promising discoveries actually do not make it into the clinic, not because they don't work, but because the development pathway and the infrastructure were actually not in place early enough. And I think that was something that we said, why is nobody picking this up? And then we thought, okay, if nobody's picking it up, then the two of us are going to do this. And that's what we would like to talk with you about today.
Yeah, absolutely. And a lot of people sort of call that the valley of death for early stage therapeutics, that, the high risk, often fatal gap between that foundational scientific discovery, the bench, and the clinical evaluation or commercialization bedside. From what you've seen, where does the process actually break down most often?
Yeah, it's usually not one big failure point, but rather a series of smaller compounding bottlenecks or gaps that are being created. Typically, we see that the valley of death is the stage when programs move from scientific validation towards development readiness. That's the stage where you have questions about the manufacturability or regulatory strategy or translational evidence that suddenly becomes very critical. And the issue is that they start looking at these at a too late time point. And that basically meaning not early enough when doing the development. So it's not the biology or the science that is wrong, but it's actually the process or the product itself that is not packaged in a way that it can actually move towards the next step. And what we often see in practice is that there is a misalignment between, let's say, the founders of a company or the scientists at a company and the investors. There is a clear path forward for both, but they're not speaking the same language. They're not having the same expectations or the understanding on how certain data needs to support, let's say, the next development step. And if you combine this also with inexperienced teams that don't really know the landscape and don't know which questions to ask or which milestones are critical, then yeah, basically these programs will lead and stall, cost more time, and in the end an investor might pull back and the product is even lost. Again, it's not really a single point where it goes wrong, but it are multiple factors, and for each individual program they're different where this will go wrong.
So ExpediteBio works specifically, I believe, in antibody and protein therapeutics. Are there additional challenges associated with biologics development compared to small molecule drugs, would you say? Debbie, would you want to start?
Yeah, indeed. So small molecules, already in the name, they are low molecular weight chemical entities and well-defined entities. And in contrast to that, antibodies are larger, complex glycoproteins, of which the function heavily depends on its correct folding, spike formation, and the range of post-translational modification, just to name a few. And this difference in complexity between small molecules and biologics also impacts the manufacturing process downstream. So where small molecules are synthesized to predictable and often reproducible chemical processes, antibodies, they are made in mammalian cell systems. And because of that, it also creates certain sensitivity to small changes in, for instance, temperature, its media composition, or even the specific cell line used already upfront. And these small variations, that can lead to significant batch-to-batch differences. And eventually to manufacturing it well, so that eventually also for the clinical trials, you have a proper defined batch. It's not only that decisions in the manufacturing process requires attention to a well-defined biological process for manufacturing. The decisions already made in the early antibody design phase are really important. That already includes, for instance, the framework selected, CDR composition, their isotypes, and that altogether can significantly impact downstream manufacturability and then scalability, which influences cost of goods. And when the manufacturing process and the protein itself is far more complex, also, and not only in structure and manufacturing, also the QC eventually becomes more complex. So we need to do far more with respect to analyzing its stability, the eventual output, homogeneity, validate functionality of the antibody in the biological assays, etc. So in essence, the biologics, they just introduce many layers compared to small molecules with respect to their structural, functional, and process-related complexity that eventually fundamentally change how to design and optimize the manufacturing process.
There is just so many different layers of complexity there, aren't there? So ExpediteBio obviously launched with this goal of lowering sort of the threshold from phase 0 to phase 1. What does that actually mean in practice? What are you doing differently to help clients with these layers of complexity?
Yeah, it means that we are trying to make the path for the first in-human study more predictable, accessible, and with accessible, I really mean that people can use a standardized approach on moving from discovery into the clinic. And we do this in a combination of multiple things within ExpediteBio. So you have the expertise of Debbie and myself, where we can help with answering certain questions. But what we are also capable of is providing innovative, but validated processes and process equipment that allow you to, with a small footprint, produce materials early on during development stages already, that will allow you to go into the clinic. And what we consider a phase 0 is a proof of concept. So basically in a hospital setting, and especially in Europe, under the guidance of a QP, treat several patients. And by generating that data, also being able then to look for further investments for the bigger phase 1 study. But by using the innovative process and the equipment that we have, we've made sure that you don't have to scale up. So when you have a good proof of concept in the clinic, you can immediately start producing your phase 1 material. And we're not, of course, doing this alone. We're just a startup company right now, but we've already have a strategic collaboration now with Sartorius that can help us with the equipment, with certain processes already, make those available to clients. But next to that, we are building an ecosystem because every project is unique. Everybody has certain different questions. Everybody has his own expertise. Debbie and I don't know everything. We know a lot, but still you have certain areas where you need to have the right expertise. And what we would do is sit down with these academic companies or startup companies and lay down the strategy, and also try to figure out which questions do they have. Do we have the right expertise in our ecosystem? Okay, then we bring those persons in, or we find the right persons that can answer those questions. And by doing that, we are the time that is required, but also the cost to actually start manufacturing these materials. So ultimately, the idea is that ExpediteBio will help these startup companies move from a scientific promise to a real world impact by using a smart strategy, but also having the access to the right infrastructure. And with the right infrastructure, it's not only for phase 0, phase 1 production, but also that you can do the phase 2, phase 3, and even commercial manufacturing. And that's also what Debbie mentioned just before this question, that it's really important to already select upfront what you want to do in the market later, because it can mean that you don't have to redesign a cell line, you don't have to redesign a process, because you already thought about what this could potentially do in the market. And that are the first questions that you need to ask yourself. And if you can't ask those questions, then ask us.
Interesting what you said about sort of building your network and ensuring you have access to the right expertise from the start, and that really can affect the process as you move forward. So when a small biotech or academic team comes to you, what tends to be the first gap that you identify? What's the most common problem people come to you with?
One of the biggest challenges we see is the absence of a truly integrated development strategy. And Richard already mentioned it before, that it's not about the science, that the scientific knowledge that the teams have. On the contrary, they have a very strong scientific mindset. They have the scientific data on the target, the biology, or even they already have a molecule. But what they are not focused on is that they have a roadmap that connects eventually their early science to eventually a development plan that regulators, manufacturers, and even investors are expecting. And also the next stages that they need to take to bring the molecule to the patients. So without that alignment of the full development plan, it becomes really hard to make the right decisions at the right time, which eventually will cost a lot of money and time. And time is money. But that's for us also why the first step is always to bring everything together into a coherent plan. So first, what needs to happen, in what order, and for which strategic reason. And the exact gaps, of course, differ from team to team, but sometimes it's their technical know-how, it's often access to the required infrastructure, and also sometimes it's simply the ability to translate the scientific data into a development pathway that is actually executable. And with ExpediteBio, we apply our Ascent model. That really means that we don't force projects into a fixed methodology, but instead we build this transformative framework that gives each project a flexible shell of expertise, exactly what already Richard mentioned, and also ensuring that all critical elements of the development are connected. And with the Accent framework, we mean that we first align, then we strategize, we create, we execute, we nurture, and eventually we deliver. I think that's really the key point what we would like to achieve within ExpediteBio.
It's not the first time I've heard time is money in the short time I've been in this industry, and I'm sure it won't be the last by any means. So if you were trying to shorten development and manufacturing timelines for a drug, where would you start? Where would be the first key thing you'd focus on to sort of have the biggest impact?
Yeah, it's already, of course, what we said. We try to align, and with aligning, we mean, okay, how can we do things smarter? So how can we put the sequences together of what needs to happen? And instead of placing everything in series, so first doing discovery, then doing some pre-development, maybe some USP developments, and then some downstream process development. No, we try to combine these and make it more overlapping and making sure that with the right infrastructure behind it, and that's having the right cell lines, but also having the right equipment, having the right assays in place. And again, the ecosystem, we are trying to shorten everything. Also, with using the platform technology that we are offering to clients, it allows them to skip scale-up issues, because the process has already been scaled. Of course, not for your specific protein. You need to do some tweaking here and there, but it's not the big scale up activities that would be required if you go from your lab to a CDMO. But also, you don't need to do the expensive engineering runs because it's a validated and proven technology. With some key steps, you can actually skip those engineering runs and far greater and more quickly produce the GMP material that you would require to go into the patients. So again, what we're trying to do is here we look at what is really needed to make products successful in the future, ask certain questions, work back to the development part, and then already start implementing that at the start of development. Sometimes people don't know, oh, do we want to do a fed batch or do I want to go to continuous perfusion? You don't have a clue maybe yet when you're doing that discovery, because the scientists are really interested in, does my protein work? Can I get the patient better? We take away the worry for them in terms of manufacturing. So we will look, okay, they don't know yet what they want to do as a commercial process. So let's have a cell line that can actually do multiple things, that you can run as a batch. You can run it in a fed batch modus, but you could also run it in a continuous perfusion. And that's coming back to one of the questions that we answered earlier. That means you don't have to do rework, meaning no loss of time, no additional cost to the investors to get the product to the market. Doing certain things early on, it gives you more flexibility that Debbie also just mentioned towards the future. And I think that's important. And that's how we try to shorten the overall development and manufacturing timelines for these protein drugs.
And Debbie, you've also been leading the Biologics workstream at Oncode Accelerator, which is trying to build infrastructure at the national level. How does that connect to what you're doing at ExpediteBio?
Yeah, that's a really nice question because it really comes together. Oncode Accelerator has the mission and vision to accelerate the development of novel cancer therapies, meaning that the different platforms and work streams and all are building infrastructure and bringing in innovative solutions to realize that ambition. So together with Professor Leuze from UMC Utrecht, we both are workstream leaders. We have the ambitions to increase the valorization of antibody therapies by bringing in deep scientific expertise from antibodies as well as the disease biology and combine that with smart infrastructures within public-private partnerships. And these partnerships are really important to achieve the bigger goal, which is bringing therapies towards the patients in need. And we do not aim for just partnerships, but building true alliances where 1 + 1 = 3 with respect to the output for our clients, which eventually means the output, in our case, is the patient. So how can we improve the treatment of patients? And in work streams, you often see that there's a lot of innovation applied at a single level. But innovation is not just about that. It's also about accelerating the whole workflow. And that comes back to what Richard just explained. It's often underestimated how significant the delay can be and inefficient spending of capital in a transition phase from having a therapeutic lead towards a manufacturable molecule or a clinical batch for first-in-human trials. So for both the investigators as the investors, it's important to navigate their programs towards the clinic, and that based on a well-defined budget and efficient route path forward. And that exactly is where ExpediteBio operates at that interface. And that comes nicely together with the mission of Oncode Accelerator. So not only as a workstream lead, but the total mission is really how can we bring novel therapies to the patients. And that all might sound really straightforward, but in practice it requires a deep expertise and strong partnerships. And where Oncode Accelerator is mainly focused on accelerating drug development for oncology, with ExpediteBio we focus on oncology and beyond, and then mainly in biologics.
As you say, partnerships are super important, especially in sort of this phase of the drug development process. As we were talking earlier about networks, building your network is super important. How should a small team approach finding the right development partners when, they maybe don't have a big network yet?
Yeah, that's a good question. We try to help them, of course, also with these type of questions. But I think that even if ExpediteBio is not at the table, I think it's most important for the companies to create clarity and really ask themselves, which questions do we have? Do we need help with the development or do we need manufacturing capacity? What are actually the questions that you have? And then from answering those questions first internally or together with ExpediteBio, then start looking, okay, what is the right expertise and what is then the network that you need to tap into? And I think that's the nice thing about the Eco Hub that we want to create as well. And we are creating. So we already have multiple players that can help us in different stages of the product lifecycle. Is that if you would do this together with ExpediteBio, we can also, if you're not, let's say, going to work with our platform, that's fine, but we can still advise you where then to go, to which CDMO or which regulatory body you should contact. And we have direct access to these people, which would save you a lot of phone calls, emailing, and trying to figure out who has the right expertise. So it's really important that you as a company first create clarity on what is your own expertise, what can we do in-house, and where do we have questions and where do we have the need for outsource help.
So what does make a good partnership in this space and what tends to go wrong?
A good partnership is that for me in practice, and I've worked at CDMOs as well, you need to provide flexibility. So if you are not capable of working together, especially with these early products, things will change, things will come up that you haven't expected. And if you are then very rigid in, let's say, just, yeah, we have this slot available, you have to manufacture now. No, let's skip that slot. Let's find a good process or do the right things that the product actually benefits towards the future. So I think that flexibility with the partners that you select is the most important thing. On the other hand, I also need to say the counter, of course. The flexibility is sometimes the death of a project, because especially if a scientist thinks, okay, let's change another thing to the protein, at a certain moment you have to stop. 80%, I always say, is good enough. The last 20% will cost you 80% of the time and money again, and it doesn't really bring you closer to the market.
Don't let the perfect be the enemy of the good enough.
Exactly.
So academic teams obviously often have very, very strong science, but then less experience navigating development. What's the most common mindset shift you need to help them make between that sort of academic mindset to then, GMP manufacturing?
And that's a really interesting point, and which I currently also experience in an ongoing project, is really to train the scientific team into what are the next steps to take into bringing their product towards the patient or the market. So the biggest shift is that it needs to change from interesting science literally to an actionable development program. And understanding that difference, that's critical. if you want the product to succeed. And again, it's of course depending on the team and the knowledge that they have. They are very knowledgeable antibody scientists that already brought a molecule to the clinic. They know their way. And there are, of course, on the other side, the teams that have a very great biology scientific mind, and they do not have experience either for bringing an antibody into a development phase. Let's even say that they can bring it to the manufacturing space. And that's exactly where we want to help those projects with bringing our knowledge and network to that scientific team. And from there, we would like to collaborate closely with the team to turn those insights into a roadmap so that all steps are aligned and translated into a development plan. And they also always have a budget that they need to work in. So we would like to create a development plan that is fitting their budget, or realize what the budget is looking like so that also the investors are aligned. And that they are not working on a development plan that they think is realistic, while eventually it costs a lot more money to bring that product to the patient. So to align even that is critical for most of those teams. So Richard already a couple of times mentioned it as well. It's not about losing the scientific depth. It's just adding the additional layers of knowledge and focus at the right time in their product life cycle to make it a successful product towards the market or the clinic.
Finally then, if the infrastructure and support structures you're building work the way you hope, at ExpediteBio and Debby with Oncode Accelerator, what does that look like in 5-10 years into the future for Dutch and European biologics development as a whole? Debby, should we start with you?
For Oncode Accelerator, I think it's really a major milestone if we can literally combine all the innovations that are present in the platforms and all the capabilities that are present in the work streams, where I'm not mainly active in the biologic work stream, but there are more work streams involved. And that together we literally show that we can reduce the failure rate of novel therapies. And with ExpediteBio, we would like to add not necessarily innovation by using an organoid or a patient-derived model, but using our infrastructure and hub to literally fast forward the molecules with the data towards actually a first-in-human study. And that's what personally I hope that together we can achieve that, because that will make a big difference in reduction of failure rate of novel therapies, but also to increase the valorization rate of all those brilliant scientific inventions that are currently stalled with small biotechs or in academics.
Richard, do you have anything to add there?
No, I think that was correctly captured by Debby. And if you just want to summarize it, the challenge is not to getting a better biology. It's building a better system to actually carry the biology forward. And that is the reason why Debby and I set up ExpediteBio. And we're lucky that we already have several collaboration partners and that we can work together with Oncode Accelerator to also achieve this. And hopefully in 5 years' time, Dora, we can sit down again and look back on what we did.
Absolutely. Yeah, I look forward to that. We'll get that in the calendar. Cool. Well, thanks a lot for your time today, Debby and Richard. It's really been great to hear from you.
Thank you.